let's go through the step-by-step formation of a red blood cell, from a stem cell to a fully mature red blood cell. The process begins with a hematopoietic stem cell, a HSC, the precursor to all blood cells. If the body needs more red blood cells, signals such as low oxygen levels known as hypoxia, the hormone erythropoietin, abbreviated EPO, and nutrient availability stimulate the stem cell to differentiate into a common myeloid progenitor. Once committed to becoming a red blood cell, this progenitor develops into a proerythroblast, marking the start of erythropoiesis. During this stage, the cell accumulates ribosomes, which are essential for protein synthesis, particularly hemoglobin production. The next stage is the basophilic erythroblast, where hemoglobin production begins, though the cell still contains a large, dense nucleus. As the cell progresses to the polychromatic erythroblast, hemoglobin synthesis increases significantly, causing the cell to take on a mixed staining pattern in the color gray-blue. The nucleus begins to shrink in preparation for removal. Next, the cell becomes a normoblast, or an orthochromatic erythroblast. At this stage, hemoglobin content is nearly fully developed, and the nucleus is expelled, making the cell enucleate for the rest of its lifespan. The final immature stage is the reticulocyte, which is released from the bone marrow into circulation. Within 1 to 2 days, it matures into a fully functional red blood cell, also known as an erythrocyte, ready to transport oxygen throughout the body.